Hey everybody, welcome back to yet another monthly journal club brought to you by Life Extension Advocacy Foundation and Lifespan.io. I'm Dr. Oliver Medvedik coming to you, as always, from New York City, Cooper Union. And we are going to be doing a journal article from the Church Lab. We've got a number of guests here joining us online. We have Dr. Christina Aguayo-Mazzucato from the Joslin Diabetes Center, and the whole panel of other folks joining in. I think there's some folks from your lab as well. Isn't that correct, Dr.
Yes,
Aguayo?
that's right.
Okay.
Yeah, thank you so much for having us.
Oh, my pleasure. The more experts, the merrier, since I'm not an expert in most of the papers that I review, although I pretend to be. And, so more eyes on the data, the better that we could dissect it. So, something a little different. I'm going to put my reading glasses on because aging catches up with everybody. And I'm going to screen share the paper that we're going to be covering right now. Hold on a second. Okay, so is that coming in pretty clearly? All right, so the title is A Single Combination Gene Therapy Treats Multiple Age-Related Diseases. This is Davidson et al., as I mentioned, from the Church Lab, among others. And this is interesting from a number of perspectives in that basically, so what they're using here is they're using an adenoviral vector, AAV, mediated gene therapy. So it's a small virus that can basically infect a number of mammalian cells. I think it preferentially targets liver cells, but it can infect a lot of cells. It's been in the news recently through a number of gene therapy trials, one of which actually reviewed a paper for a class, was the treatment of a mouse model for Duchenne muscular dystrophy, which was pretty cool. And in this paper here, what they're doing is a combination therapy. So they're using several AAV vectors to basically target several genes at once that are implicated in basically diseases of aging. And they basically want to, it's basically a proof of concept for a combinatorial type therapy, right? So if you can target several different genes at once, can you target several conditions at once? And one of the kind of reasons for doing this, and the authors mentioned this at the end of the paper, I'm gonna just stop sharing. I'm just gonna jump to the discussion and then usually I get to the discussion later. But as we've discussed many times before on this broadcast, aging is not recognized as a endpoint or a disease by the FDA, the Food and Drug Administration. So you want to target diseases of aging, and there's lots of diseases of aging. So if you can target several of them at once, the authors mention here, importantly, this strategy also presents a more attractive path toward FDA approval by focusing on the treatment of age-related diseases, which have defined quantitative endpoints. So here, potentially you can hit several different quantitative endpoints at once, and potentially, in that case, get things faster to market. So let's take a look at what they're doing here. Share my screen. Okay. So they're looking at several, so the genes that they're looking at here, and there's 3 of them, they're looking at fibroblast growth factor 21, FGF21, so they're adding in extra copies of that, alpha-klotho, and transforming growth factor beta 1. And the TGF beta 1, I believe that what they're doing here, that is, they're adding in expressing a gene that's a soluble version of the receptor, basically to knock it out its function. So I believe 2 of them are overexpressing, and 1 of them is knocking it down. So they mention here that alpha-klotho, it's a known regulator of intracellular calcium, provides protection in heart and kidney pathologies, which they'll look at later. TGF beta 1 plays an important role in age-related hypertrophic cardiomyopathy, immune recruitment, and extracellular matrix formation. And FGF21 is established in metabolism and glucose handling. So they look at several different conditions that are targeted by these genes. So again, they selected AAV1, so AAV, sorry, and they injected mice with these vectors. So we're gonna go to figure 1. And basically what they're looking at, the models that they're looking at, is essentially they're delivering these either singly or in combination, and they just abbreviate these. So FGF21, the soluble receptor of TGF-beta R2, and alpha-klotho, and they're abbreviated here TFK for the individuals and in combination. So either, TF, TK, FK, or TFK, all three together. And in figure 1, they have a systemic AAV delivery. So I'm assuming that this is an injection into a vein. And basically the first model that they're looking at is a model of obesity. So obesity is one aspect that they're looking at for treatment. There's only 4 figures, so I'm gonna just briefly mention the 4 conditions that they're looking at. Figure 2, and then I'm gonna scroll back, is looking at, they're basically looking at in a diabetes model. So they're looking at, for I believe type 2 diabetes, I'm looking at various, glucose and insulin response parameters in mice. And this is mice on a high-fat diet. And in figure 3, they're looking at regeneration of the kidney function. And this is a model. So this one is a simulation of kidney atrophy using something called unilateral urethral obstruction. So UUO. So this is basically a surgical intervention, which they basically just tie a ligature somewhere on the kidneys to essentially cause them to malfunction or atrophy. And they're looking at to see, the level of regeneration after the AAV vector treatment. And the last thing they're looking at is a cardiac condition. So in this case here, what they're looking at is they're also using some sort of a ligature around the aorta, and they're basically looking at improvement of cardiac function after delivery of these vectors. So let's just keep those in mind, because the authors claim that all of these are sort of correlated with aging phenotypes. Clearly, problems with glucose regulation, problems with weight, problems with renal function and heart function are in cardiovascular function in general are conditions that increase negatively with age, and these are all conditions that the authors want to see if in single treatment or in combination they can basically treat. So in figure 1, what they're looking at here is basically weight loss. And they're looking at essentially weight gain and days post-treatment using the AAV delivery. And basically the bottom line of this figure, actually the bottom line of figure 1 and 2, is that only one of the treatments really has a major effect, and that is the FGF21. So that's in the red line here. So basically the control is basically normal diet, and control high-fat diet. So normal diet has a low weight gain down here, dotted line from 0 to 100 days. High-fat diet up here is the weight gain in grams for a mouse. And here are the various treatments. And you can see that the one that's the most obvious effect here is the FGF21 that's delivered by AAV. Over a period of time, maybe a slight increase. I don't know if that's statistically relevant. They have FGF21 in combination with klotho. I think they mentioned that wasn't that statistically significant. Some of the other interventions did not work as expected. Alpha klotho, for example. A similar effect is seen with long-term effects of FGF21. So they just focused in this condition here for FGF21. And the take-home message essentially was, and you'll see the same thing for figure 2, so it wasn't that extraordinary, but essentially for weight gain as a result of high-fat diet, and also looking at the glucose levels in the blood of these mice, and first using a glucose tolerance test and an insulin tolerance test. Stop sharing here for a moment since I'm focusing on here too much. really most of the effects were due to an overexpression of FGF21, but weren't really, there wasn't that much of an additive effect through the other genes. We'll see that changes later on with their models for renal function and heart function, where you get an additive effect by adding in an expression of one of the other gene constituents, but not for high-fat diet yet, or not for not for glucose levels in the blood. So, are there any comments so far? Should I put, if anybody wants to take a look at the figure and look at the data, I'll just put this back up.
We've got nothing on social media yet.
Nothing on social media.
Not yet, but if anybody does have any comments or questions, obviously pipe up on the channel in Facebook there, where we are watching, as they say.
Okay. So I've scrolled to figure 2, where the figure is basically looking at systemic AAV delivery of combination gene therapy, reversing symptoms of diabetes for mice on a high-fat diet. So we have folks that study here diabetes. And so if there's anything that pops out in this data that you'd like to comment on, because just looking at a glucose tolerance test, which is basically the clearance of glucose in the blood, or insulin tolerance test, which is an injection of insulin and basically its responsiveness to clearing glucose in the blood, you could see, and this is basically the area under the curve. So essentially, a translation of this data, figure A and B go together and C and D go together. You can essentially see that, again, just very similar to the high-fat diet mice and the weight gain or the weight loss, the most pronounced effect was with the basically FGF21. You didn't really, in combination, yeah, you get an effect too, but clearly it's attributable primarily to FGF21. So so far in this data, we're not getting really any additive or synergistic effect that from any of the other genes that they're testing, but we will see that shortly with the model for renal failure. We have something coming in right now. Let's see, click on this chat. How good an analog are mice fed in high-fat diet to actual type 2 diabetes? So yeah, how good is an analog to type 2 diabetes for mice fed a high-fat diet? Obviously, these are all models, so there's going to be pluses or minuses. Is that a decent analog for anybody out there?
So I think we also use high-fat diet as a precipitator of diabetes. And there the main mechanism is mainly enhancing insulin resistance, which if sustained long enough will lead to beta cell failure, and then you will get over type 2 diabetes. And that's something they're trying to evaluate here. And as you were going through the figures, I think for us, whenever we see these complex models of high-fat diet and you have effects in so many tissues, it's important to dissect the effects on peripheral insulin resistance or fat mainly, and then the effects on beta cells, which would be their HOMA-B. So it seems that these effects, the main benefit of FGF21, as you were saying, seems to be on reducing peripheral insulin resistance, which would be mainly on the fat. Because if you look at the HOMA-IR, you have a parallel decrease of HOMA-IR and then an improvement of on HOMA-B. So it seems that their beneficial effects within this type 2 diabetes model is mainly on the adipose tissue. And that's always something when we're trying to figure out how beneficial a treatment is. One of the first questions is always, where is the effect? Is it peripheral insulin resistance or is it beta cell function? So that would be my reflection on these results.
So having those HOMA results actually is I'm just going to go back to the figure here. So that's very promising, the data that you see here.
Well, what it would mainly be saying is that these effects are mainly on peripheral insulin resistance rather than insulin secretion itself. It seems that the benefits are by decreasing peripheral insulin resistance, which they don't show. They show weight or lack of weight gain. So that could be just because these mice don't have as much fat accumulation.
Right.
So that by itself just will compensate
right.
For that.
Right. Okay. So just as you mentioned that, so as a control in figure 1, they mentioned that they're losing the weight. Primarily, it's a weight loss gain through adipose tissue, muscle mass, and bone density haven't been affected. So that's something they looked at as well. So far we have FGF21 doing most of the heavy lifting here, if not all of it, for at least weight gain and glucose levels in the blood. They have 2 other models here which they jumped into, and again, I don't know how accurate these are to reflect kidney failure and renal fibrosis, because that's something they mentioned is a problem with the aging community in the world. But this disease model is something used in mice, and basically, like I mentioned, it's tying a ligature around the urethra, or basically preventing urine from exiting the kidney, from my understanding, and that causes a backlog of urine and toxicity to the kidney, and sounds kind of painful to the mouse, but basically you get atrophy of tissues in the kidney, right? So that's seems a very crude but direct way to induce renal failure. And what they see here, so these are basically cross sections of kidneys, and they use something called Masson's trichome stain, which basically stains extracellular matrix and cells, so you can basically get see the mass of the kidney. So this is basically a kidney from a mouse. This is a control day 0, and this is a week after this tying of this ligature around the urethra. So you can see that there's quite a lot of atrophy here within the kidney, and this is clotho, and this is FGF21, and this is both together. So you have a just by and this is the percent atrophy. That's basically a decrease through a combination of the 2, which is designated as significant. Even just by eye, if this is a representation, it's clear that that's pretty impressive, at least 7 days. So this is the model that they have. This here is just a stain, and they're looking at, I believe, smooth muscle, alpha smooth muscle actin, which is basically so they basically mention here that myofibroblasts are key mediators of extracellular matrix formation and express alpha smooth muscle actin. So if you basically have more damage occurring, you're going to get more of this deposited, basically more ECM, less cells. That's the red here. And as an example, they just show you the AAV with the alpha clotho that's expressed. Again, this is looking at all the different gene variations through the AAV vector, percent smooth muscle. So the percentage is alpha smooth muscle, the percentage is higher for control, less for everything else, and the least for TGF21 in combination with TGF beta receptor 2 ablation in combination with FGF21. So this is the first time so far in this paper where they show that having a combination of 2 genes, so basically knocking down TGF beta receptor 2 and increasing FGF21 seems to have a beneficial effect in this model of renal failure. The issue I have here is, how translatable is that to an actual aging model? Because you're tying a ligature around a ureter in a mouse and seeing, essentially cells dying off, and you see a prevention of that when you have these two genes inserted via AAV. So if we go back to, if we just scroll back right now to the point that the authors were making, I think I'm still sharing. Let's share the screen here. If I scroll back to the point that the authors are making back here, so we so far know that FGF21 is the sole gene that's required, or overexpression, and I don't know what levels they're overexpressing it. I'd have to take a look at the supplemental data. We see the strongest response with just FGF21, but we see a similar response with the two genes, so I'm just going to abbreviate it TF versus just F. So having either TF or F basically gives you a comparable, comparable rescue when it comes to an insulin tolerance test, a glucose tolerance test, and it also seems to give you a comparable, if I'm not mistaken, so that's orange, it's down here, a comparable or nearly comparable effect when it comes to weight loss in a high-fat diet. So at this point in the author's paper, they can, legitimately claim that at least for these, you have two distinct models of, of something going awry here. Basically, renal failure that's unrelated to the high-fat diet, right? And then you've got, conditions arising from the high-fat diet, and using both of those genes in an AAVS1 vector system. I think it's probably two separate AAVS1 vectors, because I think, AAV, because I think AAV basically doesn't have that much space as a vector to jam things in. I think it's like maybe like 5 kilobases or something like that, information you can put in it. But so far, by the time we get to figure 3, you have you have a combination of at least the proof of concept is that you can hit two different conditions at once using this AAV vector system in one mouse. Although I'm not sure if this is the same mouse, so that's something I have to dig into, or if this is actually two different mice. I don't think these are a mice from the same high-fat diet, if I'm not mistaken here. So if they're not, my question would be, would it also work if you had a high-fat diet mouse also undergoing renal failure? Would you need to change the concentrations of the vectors? I'm not sure. Maybe somebody can comment on that. Any thoughts, comments so far? No. All right. So let's share the paper. So we're down to the last figure, actually. And this figure here is similar, well, not similar, but kind of similar, I guess, surgically to the renal failure model, in that they basically use a ligature. I think they use some kind of a stent or something to basically constrict the aorta. And you're also essentially putting in, stressing the heart, if you will, by having this sort of ligature. So this is called the AAC mouse model. It's aortic. I think it's aortic aneurysm constriction. I could be garbling that completely, but basically it's using some kind of a stricture around the aorta, and they're doing electrocardiograms here to look at several parameters, just like they looked at renal function. They're looking at something called fractional shortening, which is basically a functionality of how essentially, I believe, the heart tissue shortens when it beats, and ejection fraction, essentially how much blood you're getting ejected from the heart. And what initially they show here is these are from echocardiogram readings, which is basically like a sonogram. And day 0 is basically, this is when they're either right before or right after they add the stricture. So you don't really notice any difference in fractional shortening or ejection fraction if you add these vectors. So these vectors have been added, but the ligature hasn't had time to kind of do its damage yet. And so the baseline is about the same. So for both ejection fraction and fractional shortening. So these are several different mice. So C for control. And then the various genes here. I have to remind myself what the acronyms keep meaning. I think it's TGF beta receptor 2, soluble TGF beta receptor 2. Same thing with Klotho, and the same thing with FGF21, and then a triple combination. I think what they notice here, though, is that they start seeing, and they don't really have an explanation for this, but they start seeing negative effects if they have, I believe, a triple combination. So if over a period of time, so you start seeing a trend basically going up from left to right. So you want your fractional shortening to increase and your ejection fraction to increase. And you certainly see that with, so, both the TF, which is the orange, that's the combination that worked previously in the other three figures that we saw. You see a similar effect with T and Klotho, but I believe the effect is not as, for fractional shortening, is not as pronounced, or starts to kind of drop off for TF and Klotho, and I'm not sure why that's the case. They mention here that fibrosis in the heart, which is essentially scarring, goes up higher with TFK, similar to control, and that the best response is with the combination of TF and TK. So again, the only comment I would say for this thus far is that the one thing that the authors have shown is that having a combination might be effective for treating several conditions that that affect an aging population at once simultaneously. I'm not the expert in translating these models to an actual aging condition. So again, this is somewhat artificial. You're putting a ligature around your urethra, you're putting a stricture or a ligature around the aorta, and you're basically putting stress on the heart, and it's done for a short period of time. And also, I'm not sure if these mice, they've all been individually stressed, right? So we're looking at several conditions, but they're not all happening at once in one mouse, right? From my understanding. If somebody's read this paper a little bit deeper, maybe they can correct me if I'm wrong, but I think these are at least for the high-fat diet mice, I think the figure 1 and 2 might pertain to the same mice. The figure 3 and 4, with the heart condition, the artificially induced heart condition, and the artificially induced renal failure, those are two other mouse models that are not suffering from also a high-fat diet. So take that as you will. Maybe we could, I would say that the next step, at least for me, would be to look at this in other models. Look at this in an aged mouse, or maybe a hyper-aged mouse, maybe a mouse that's basically suffering from a accelerated aging phenotype, and look at several conditions at once. In an accelerated aging phenotype mouse, would you improve, would you decrease cardiomyopathies at the same time reducing the blood glucose levels, and at the same time improving renal failure all at once, in that sort of model? Maybe that would be a little bit more closer, to an aging mouse model. I think this is a step in the right direction. The authors do mention another thing in this paper, where they're basically using a combinatorial approach to a gene therapy. That's something that we're seeing also with drug models right now in aging, and we've covered a number of papers where you're doing a combinatorial approach to basically target several pathways at once, and seeing an additive or synergistic effect. This isn't necessarily an additive or synergistic effect. This is more parallel conditions being hit all at once. Maybe not quite parallel. Like I mentioned, these aren't, all of these conditions, the caveat here is they're not all happening in the same mouse at the same time. So that would be my caveat here. Anybody have any comments as far as my summary of this?
It was very good. Where do I sign up?
Well, you know, I think the AAV viral, you know, therapy model is very promising, at least for some, definitely for some of the congenital diseases that that have been tackled in mouse models. Certainly for DMD, Duchenne muscular dystrophy, that was a very intriguing paper where they basically changed the splice variant, you know, by using the vector to deliver. Basically, I believe it was a, you know, they used a CRISPR-Cas9 based system to alter the splicing and got a functional dystrophin gene. So using this vector, I think, is very promising. I think I don't know what other things they can be targeting here. My thing, if I were to sort of go ahead with jumping ahead from this paper here to propose more experiments, I think I you know, kind of generally proposed, you know, looking at other mouse models that might incorporate you know, multifactorial disease model all in one mouse, right? So that would be so yeah, senescent cells definitely. Using AAVS vector to essentially, you know, maybe using a CRISPR-Cas9 system to recapitulate mutations that are known to promote longevity in an adult mouse, right? So that would be another series of experiments that could be done. Ablating senescent cells, for example. But yeah, I would be interested to see this, you know, maybe not the, maybe starting with the double gene combination that they teased apart in this paper in a different mouse model that incorporates several diseases at once, like a mouse that's very aged or maybe has an accelerated aging phenotype.
Well, I wouldn't use progeric mice, Oliver, to be fair, because a lot of people criticize them because they, you know what they'll say? Well, it's not actual aging though, is it? So I would probably, a mouse of choice, I would get a good old black 6 and I would try and acquire already aged black 6, like 18, 22 months old, something like that.
Yeah, but tying a ligature around a aorta isn't really heart disease either.
Yeah, I know, but it's a common criticism, isn't it? Everyone always goes, yeah, but it's not actual aging, is it? Well,
yeah, you do the best you can with the models you have.
You
right?
do.
So yeah, I mean, so I would say that, you know, those are the positives that I just mentioned about the paper and also the criticisms. Anybody have any other jumping off points for experiments in this vein? No pun intended.
Oh dear, in this vein.
So I would only say that for FGF21 at least, there's the possibility of just raising plasma levels. And I believe there's already some clinical trials, both in the setting of metabolic disease and renal fibrosis. So at least for that factor, I think it wouldn't be necessary to use a viral vector, which would be an advantage thinking of translation.
So you're thinking about just injecting FGF21 directly?
Yes.
Purified. Okay.
Right.
Yeah, that's actually I don't know how. I guess it's cost effective at the moment, at least for mouse trials, to buy and use purified FGF21. I'm assuming, I don't know.
Right, and also going forward, if it doesn't have to be gene therapy, I think it would really make it easier just in terms of translating it to a clinical scenario, if it were to get to that point.
Sorry. Yeah, I agree with that. Having a biologic that's not a not something that's integrating into a cell is probably going to be a much lower hurdle to overcome, especially if you can inject, this dominant negative version of the receptor that they used here, this soluble TGF-beta receptor 2 in combination with the FGF21, which I think that was the combination that seemed to be the most promising in this case. So if that's cost effective, that would be another jumping off point for an experimental trial, and injecting that into aged mice and seeing and looking at several different comorbidities, so to speak, in one mouse model.
Right.
Because right now we're looking at sort of 3 different mice models. Like I said, I kind of looked through these, been looking through this materials and methods section, and these are C57 black 6 mice, and these are aortic constriction model. So these are they're different mouse models, right? So the first 2 figures are one mouse model, and then the renal is another mouse model, and then the heart is another mouse model, and then they're saying that they could potentially be combined, right? Because
Right.
the whole gist is that these are all conditions, these comorbidities all exist in one person. So the next obvious step would be to look at a model that has all 3 of these conditions, or all 4 of these conditions, or maybe additional conditions that are happen with aging, and see if they basically if they can all work together in the same organism at once. So
Right, and I think something you brought up is the nephropathy associated with aging or with diabetes is not necessarily the same type as the one induced by blocking the urethra. So they might be physiologically different entities.
Right.
And as you were mentioning, a model that is more physiological in terms of aging or diabetes and its complications would be more biologically translatable.
Right, because I'm looking at for the urethral obstruction. I'm not sure if those were aged mice, because obviously the older the mouse is, the worse the recovery is going to be. So with this type of ligature or recovery thereof, would that work as well on an older mouse as it would on a with this combination gene therapy or injecting these factors, would it have as much of an effect? I would probably guess no. Or would there be any effect for a mouse that's post middle age for a mouse? I'm not sure. But I do think the data thus far is, as they say, promising, and I think it opens up the avenue for further experiments to be done. Some of which that we've mentioned, that we can employ in a follow-up paper, and maybe the authors are already working on these experiments. I think
It's George Church, of course he is. He's got about 400 things going on at once. Yeah,
so if he hasn't thought of these experiments, George, I'm just giving you some ideas here, free of charge. So.
And he's got the mammoths upstairs, but you're not allowed to see those. That's in a secret area of the lab. That's where they keep the mammoths and dinosaurs.
The mammoths and dinosaurs. Yeah. Well. Yeah.
Well, he did say that he wanted to bring the woolly mammoth back, didn't he?
Yes, he did multiple times. Yeah.
He would do it. I think he would do it. But you just know that someone's going to quote that thing out of Jurassic Park. I think they already have.
Oh, of course. Anyway, so that is the paper. That was a somewhat brief summary, a little better than a blog and an abstract not as deep as me going through every single subfigure, but somewhere in between.
Ah, there you go.
I don't know. Did you blog about this paper, Steve?
Of course I did.
Okay, you blog about everything.
I
You
try.
do an excellent job blogging about this stuff.
I do my best, but there is, to be fair, there is so much going on in the field. It's very hard to actually keep up with everything. It's crazy.
Yeah.
I've got a human trial to write about tomorrow about rapamycin and skin aging. So there's been a small trial of that, and the results look interesting. But yeah, it's absolutely crazy what's going on.
So Marcos Rios, who I believe does the journal club at GenSpace, who's just started a aging journal club there. So welcome, Marcos. What percentage of cells does the AAV model successfully introduce the genes? That's a great question. It's not 100%, obviously. I'm just, it's probably some buried somewhere in the supplemental here, but I think based on when I read the DMD paper, I think some cells, like intestinal cells, were not infected at all. Liver cells were very well infected. I think muscle cells were also pretty well infected, but it wasn't like 100%, infectivity. Obviously, it also depends on the amount of vector that you use. So off the top of my head, I can't tell you what percentage, but it is far, far from 90% or 100%. So a lot of conditions, like for example, Duchenne muscular dystrophy, and probably some of these conditions that were studied here in this paper, like tissue regeneration for kidneys and heart, and also for lowering of blood glucose levels, you don't, it's not an all or nothing response. So if you get some percentage improvement, it's better than no percentage of improvement. So so you do get some amelioration that's measurable. But I can't give you a number here. I would have to look that up. It's just it's definitely far from 100%. And again, I think there is some tissue preference for the AAV vector. If I'm not mistaken, I think liver cells get preferentially targeted, although muscle cells also get targeted. Steve Hill, one of the issues of the AAV is the massive cost, and costs upwards of $1 million to produce enough vectors for a person. Accessibility a problem. How can we make this technology more scalable? I don't know. These numbers that I read, like $1 million or $300,000 for a gene therapy, or for that matter, $6,000 for a generic shot of epinephrine. I mean, are these real numbers, or is our just economic system all upside down, sideways, and people can just throw on whatever number they want? Does it really cost $1 million to make AAV1, or is that just what some entity has arbitrarily said? Let's make it $1 million because insurance companies will pay it. But hey, it only costs us 5 cents for every $10,000 we bill. I don't know.
I mean, I know George Church was hoping to make it scalable in the manner that he's done with the sequencing, because of course he was involved in he was one of the founders of the Human Genome Project, and look how many times he's innovated in that and reduced the prices of sequencing,
right?
Plus hundreds of thousands, and take you like a couple of days to sequence something, and you can now have it done for the price of a couple of $100.
Well, I'll be a jaded skeptic here and say that if you needed a doctor's prescription to sequence your plasmid, it would probably cost you $500,000 to sequence a plasmid.
Yeah, I mean there is something wrong with it, but that is the going sort of cost if you're going to produce them at GMP.
Yeah. Well, I mean, again, it'd be interesting to have a discussion where we can get somebody who's really in the know on this to see where in the supply chain the costs actually kick in. There was a scandalous report that just came out it's been coming out for insulin, for example, which is used to treat type 1 diabetes. And I don't know, maybe some of the folks here on the panel could weigh in on this, but recombinant insulin has been around since the early 80s. That's kind of launched the biotechnology revolution. And why it doesn't cost as cheap as Tylenol, I have no idea. It's not because it's that hard to make, my understanding. It's just because that's how the market is structured to, you know, to kind of wring that kind of level of profit out of certain things. And I don't know where in the supply chain the costs get astronomical, right? So it would be very interesting to see that supply line and see, you know, where the costs come in. You know, it's like, oh, it costs 5 cents here, 10 cents here, $1 here. Whoa, it goes up to $10 million. Like, where did that where did that transition point, inflection point in getting into astronomical costs come in? I don't know.
And I don't think it's the same across all countries, at least for insulin, which is one of the criticisms. And I think the American Diabetes Association and other advocacy groups are really looking into the accessibility of this and the impact on the quality of life for patients with type 1 diabetes, because some of these patients are really highly impacted and they have to travel to find affordable insulin. I completely agree with what you're saying.
Because I saw an article written where there was a positive spin being put on biohacking, where some people are basically trying to make insulin in their garage, and they're like, wow, isn't this cool? I'm like, no, that's not cool. Something's broken if you are considering strongly to synthesize insulin in your garage, and considering that this has been manufactured since the early 1980s. I mean, something is really broken if in 2019 we have to resort to garage manufacture of insulin. I don't know, at least in this country. So it's literally come to the conclusion where, yes, there are people running clandestine laboratories, not clandestine, but DIY laboratories to try to produce insulin in their homes. And, you know, to me, that's a symptom of something really broken. If that's the solution, or the perceived solution to the insulin supply problem. And like you said, I mean, this might be just a quirk of the US based system of drug pricing and basically how things get to market. I don't have a solution. Well, I have some potential ideas for solutions, but
I do.
Yeah.
Socialized healthcare, my friend. Yeah. This is what we've got in the UK. I don't want to get on my soapbox here, guys, and get too political, but being in the UK, I'm so glad that if I ever did develop diabetes, which is a distinct possibility as it runs on both sides of my family, I wouldn't need to, you know, panic. I could just go to see my my doctor, and I would get easy access to insulin, no problem, or depending on obviously how severe it was. I mean, it could be dietary controllable diabetes like my dad had, but my, you know, just varies.
Yeah. That's a solution. The problem, the problem is, as always, implementation, right? That's the problem.
Well, no, it's easy for me to say because obviously we have that system here, but swapping, you know, I really don't have all the answers, but it seems to work quite well here.
Yeah. Well, anyway, that would be a fertile topic for discussion.
Oh yes.
For later, because like I said, the system is not completely bankrupt, but it is hobbling at very low efficiency, and it needs to be improved. The scientists are doing a great job. Something's happening at the far end of translation into the consumer. That's a problem at that end.
Well, you know, I think the obvious solution would be to come up with permanent solutions to metabolic disorders like diabetes, and then we can just cut the profiteers out of it, can't we?
Yeah, you're going to always need a mixed solution, mixed therapies, obviously, because of people having congenital problems and so on and so forth. But anyway, I think I've wrung out as much information as I can from this paper. I think it's very promising, but I think there's a lot more that could be done with the data that's presented here. And I think some of the suggestions we made, you know, I hope they bear out in upcoming papers that tie into this paper. I think it would be really cool to see these kind of parallel avenues take place, which is combination pharmacologic interventions and combination gene therapy, or, you know, biologic interventions, basically looking at comorbidities, because, you know, if the FDA won't recognize aging as a disease, well, then maybe we'd need to come up with some sort of comorbidity model that makes the doctors happy, as far as looking at several measurements at once, and call that not aging, but call that something else, right? Multifactorial disease condition. There you go.
You mean
That's
a syndrome? Aging is a syndrome.
Yeah.
I've said it before, and I still think aging is best described as a syndrome because it's a group of symptoms that are commonly associated with each other. If you want to be twee about it.
But anyway, hopefully, you know, hopefully these interventions will, when they hit the market, we'll figure out the other problems, because if we can't price insulin, which is a drug that's really older, at least the recombinant version is older than most of my undergraduate students, or all of them. If we can't fix that supply problem, then I have very little optimism that a multimodal approach is going to ever hit the market. So a lot of work to be done on both fronts.
It'll be all right once the patent runs out and it goes generic.
Yeah.
That'll solve your problem.
Well, here's the problems with that. Epinephrine is generic too, and yeah. Anyway, sorry to end that on a low note, folks. Yeah. But that's meant to be that was also meant to stimulate people to think about the supply problems that we have. And, you know, if you're a researcher out there, get involved at that end of the spectrum, because, you know, if you're making some really important discoveries, as much work as that is, the job's not done yet. The whole point is to actually get this to people where it matters. So you need to really consider the entire chain of events, you know, as far as the therapeutic pipeline is concerned. Maybe that's too much to ask for researchers who are already bogged down with the minutia of day-to-day research. But it's something to keep in the back of your mind, at least.
Well, that's good. At least you managed to pull that back there from the precipice of the down note. So kudos to you, Oliver. We ended on a slightly more positive note. That's great. So yeah, but as I say, socialized healthcare, that's a whole nother topic. We could probably get into that, and the manufacturing cost, that might make an interesting journal club. We can get people like Kelsey and that who work in pharma to come and give us some insights. I'm sure he'd be up for that as well.
Yeah, and also a you know, I mean, one, you know, a comparative I guess a comparative medicine approach around the world, right? Because it was just mentioned, obviously the US it's one of the biggest markets, but it's not the only market, and it's not the only way things are done. I'd be interested to see how things are done in Switzerland or the EU or China versus the US or Australia, or, you know, or the UK. And basically, can we compare the pluses and minuses of all of these different ways of getting things through the pipeline, and see if we can pick the best out of everything and come up with the best approach? I don't know. I'm sure people have done this, and I'd like to learn more about it, because that's a weakness in my skill set, my repertoire of knowledge.
And we can talk about medical tourism as well, which is a big thing in the community. Apparently, they jetting off to exotic locations to get these cutting edge therapies, which are not available here. I've certainly got an opinion on that, but it's probably not one that's going to make me any friends. But there we go. That's a story for another time.
Okay.
All right. So that's that then. If anybody,
Yeah, I think
unless
so.
anybody's got anything to add. No, we're all quiet on the Western Front. It's all quiet on Facebook. We've had a couple of comments.
Did we pick a journal for December?
We
Not
haven't,
yet.
have we? No, we haven't. We've got a few papers for consideration, so open to ideas.
All right.
So yeah. So anybody out there who's watching as well, you're more than welcome to suggest papers to us, as is anyone here, and we'll have a look. We're always up for something interesting, but there's always something going on, so I don't think we're going to have a shortage of choices. So yeah.
I think the recent Maria Blasco paper on the long telomeres, the mice with the long telomeres, I think that was a very interesting paper.
Yeah, all right. Yeah, that would be very interesting. I have not read that one yet.
That's the one with the super long telomeres, and they found that they age slower, right? Well, that's no surprise, because the paper from earlier this year showed that how telomeres, or sorry, rate of telomere loss is what dictates lifespan across loads of different species. It's not the length, it's the speed. So
well, anyway, let's put that one onto the pile. Because I know I proposed a couple other ones, but let's take a look at our pile of papers to choose from for next month. And no doubt what'll happen is in about a week or so, another fantastic paper will get published, and all those papers will go off the table, and we'll pick that one, and we'll run with that one. Yeah, but that's okay. That's okay. That's okay. All the other ones that we don't cover, I'll leave it up to Steve to blog about, and that'll fill in all of everybody's knowledge gaps right there.
Well, you know, I'll do what I can, Oliver. No promises, but I'll do what I can. I'll keep the presses rolling.
Cool.
All right. So I guess we'll see you. I think we've got a tentative date for the 17th of December, as I recall, for the next Journal Club. We get this one in a little bit early because, well, Christmas, and even Leaf takes a break. So we have to get some R&R in, don't we, over the holidays?
Yep. And
we'll be back in the new year, of course, as always. So we'll see you all on the 17th. Thanks to everybody that joined us today, and also everybody out on Facebook watching us live. And we will see you next time, hopefully. And thanks, as always, to the Lifespan Heroes, monthly patrons who help us make programs like this and lots of other things like the X10 show and all just lots of things. So big kudos to them. Thumbs up. Thanks for helping us out. And if you'd like to learn a little bit more about that, you can visit lifespan.io/heroes. That's it. We'll see you next time.
Take care, everybody. Thanks.
See you next time. Bye-bye.
Bye so much. Thank you so much for having us.
Oh, thanks for joining.
Yeah.